So hopefully everyone's okay with that. Okay, well, we'll go ahead and without further ado, get started. My name is Romy Amaro, and I am super happy and very excited to welcome all of you to the thesis defense of Abigail Domer, or we know her as Abby. Abby, you're in for a treat today. So Abby has had a really, I think, interesting background. Abby, did you grow up in Ohio?
I did, yes.
Okay. So she grew up in Ohio and she went to Wash U University in St. Louis, very close to where I grew up, where she received her bachelor's degree in chemistry and a minor in classics, which turns out to be quite convenient now because she knows the entire Greek alphabet, which is turning out to be very important, as well as how to say all those letters like Omicron and so forth. She then came to graduate school, actually first in 2014 for a few years, earned a master's, or a couple of years, earned a master's degree, and then took a little bit of a short break, I would say, where she went off and she taught at a couple of different institutions. I think, was it Mesa College?
Mesa and Palomar. And I taught high school for a year. Mm-hmm.
That's awesome. And then she decided to come back, and I'll never forget, Jamie Schiffer is somewhere on this call. And Jamie was a more senior grad student at the time, working in case, and which I'll tell you more about. And she said, there's this really good student. Her name is Abby Domer. And I think, we probably want to take her. And I hadn't interacted too much with Abby, but coming a recommendation like that from Jamie, I thought, okay, we'll see how it goes. And so this turned out to be really interesting because together, the two of them brought this wonderful world of aerosols into my group, which I had never in my wildest dreams did I ever think I would be interested in atmospheric chemistry. I was someone who came from really sort of the more human biology side, cell biology, molecular biology. And but what turned out to be so interesting was that, as Abby's going to tell you about, was that they had discovered that these bits of the ocean were being aerosolized, and they weren't just salty water, but that they actually had biological material in them. So we set out on maybe what we thought at the time was a little bit of a crazy idea to try to bring the tools of computational biophysics into more of the world of atmospheric chemistry at this wonderful intersection that certainly we had never traveled down. And it was really sort of a trip, Abby. I think the whole thing from start to finish was really fantastic. And Abby has been so sort of brave in what she's been able to do, and not only courageous, but also just so phenomenally technically talented and just so brilliant. So, she has something like 18 publications, and actually, I should say, gosh, I'm like getting flustered because there's just so much wonderful things to say about Abby. So I don't even almost totally know what to say, but I want to show you this. This is sort of very unusual that just today. So Abby's one of the few people who has had her work featured in the New York Times. And I don't know if you all can see this. Are you seeing the right screen, or do I need to swap displays?
We see it.
Okay. So this was last year, and this was this morning. So last year, we started pivoting to work in coronavirus. She's going to tell you all about that. And this really beautiful image that she created was the first simulation of the SARS-CoV-2 virus, set a new high watermark for computational virology at about 305 million atoms. She's going to tell you about it, but was so phenomenal that Carl Zimmer, featured it in this really beautiful piece talking about how scientists all around the world really came to the fore in the fight of coronavirus. And then she continued on this work, and really then it became truly intersectional, I think, sort of uniting her love with aerosol chemistry, but also now with studies of the virus, and was able to build and simulate after many years of tool building and trials, the coronavirus in an aerosol. So this is just today. So as Gil was mentioning, if you happen to get the New York Times, take out the science section, and you'll see her work featured there, which has just been really such an incredible treat. So I guess I should stop talking and let her tell you all about her amazing work. Sit back and relax. I think you're all in for a remarkable journey and treat here as we listen to Abby's defense. Oh, and so I'll just say just some sort of formalities. We're going to let Abby give her talk, and then I will open the questions for the general audience, and then the committee will break off with Abby, and we will continue to ask her questions, and then we'll reunite afterwards with the group. Okay. So Abby, would you like to take it away?
Of course.
I could go on, but really we should let the stars speak. Okay.
We're good. I'm gonna go ahead and share my screen. Let's see.
What a beautiful child.
Optimize for video clip. Okay. Share. So you should see a virus particle in the background. Yes.
You do.
And if I do this, you should now see my slides. Is that correct?
Yes, we do.
Okay, awesome. So like Romy said, I have so much to tell you about. So just, buckle up. We're going to go a little bit quickly over we're going to touch on a little bit of everything that I've been working on. So we're going to start with my work on sea spray aerosols, and then I'm going to move into some of our more recent efforts to understand the coronavirus behind the COVID-19 pandemic. So my talk today begins and ends with aerosols, and I'm not just referring to the stuff that comes out of your hairspray cans. Aerosols are tiny solid or liquid particles with natural or anthropogenic origins that get suspended in the atmosphere and can even travel across the planet. So with the rise in awareness that COVID-19 is largely spread through airborne transmission, understanding aerosols has become increasingly important, not just for environmental and climate reasons, but also for public health and safety. From an environmental perspective, aerosols represent one of the largest sources of uncertainty in our climate models. So you can see here on the left, this figure from the IPCC report shows that aerosols can largely offset the global warming caused by greenhouse gases, but the error bars here are just so massive. And this really impacts the sensitivity of our climate models to things like human-driven climate change, and also affects our ability to advise policymakers. So as Romy mentioned, I work for the Center for Aerosol Impacts on Chemistry of the Environment, led by Drs. Kim Prather and Vicky Gracian. And our main focus has really been on sea spray. And that's because sea spray makes up a significant portion of the natural aerosol budget. But it's also, they're also some of the most complex and least understood characters in atmospheric chemistry. So the cool thing about CASE is that the center is known for bringing the ocean into the laboratory. And this is really what got me hooked on sea spray research. We're actually able to siphon real ocean water from the ocean just off the coast over here in California into tanks and then spike it with nutrients and induce a phytoplankton bloom just like the ones in the ocean. And we can also recreate mechanically the same bubble size distribution that's created naturally by breaking waves, and then collect the sea spray that comes off of these breaking waves to study it in a controlled laboratory setting. So all of these experimental scientists are our collaborators, and it's been really cool to work with experimentalists studying this stuff and match up, our simulations with their experimental observations. So the first part of my work is going to focus on these aerosols. So here's an outline of my full dissertation. It's titled From Viruses to Sea Spray, Applications of All Atom Molecular Dynamic Simulations to Environmental and Biological Aerosol Systems. It's divided into 3 parts. Part 1 is the study of the air-seawater interface. And understanding this interface is so crucial because it's the first point of contact of sea spray with the atmosphere. The interface really regulates interactions with sunlight, and it regulates the reactive uptake of atmospheric gases and the diffusion of water, and it can even template the formation of ice crystals. So we need to make sure that we understand this interface on a very fundamental level before we can scale up to bigger systems and higher complexity. In part 2, we try to understand the aerosol from a more whole particle level by really ramping up the size and complexity of our systems. So here I'm going to talk about what I consider the real crux of my research, in which I'm able to build and simulate whole sea spray aerosols and then evaluate their molecular level properties. And then finally, in part 3, I show how we're able to apply the lessons we learned from simulating sea spray to tackling ultra large systems, and specifically the SARS-CoV-2 virus and the role that respiratory aerosols play in its stability and transmissibility. So just as a brief introduction to the methods that I use, I'm a computational chemist, so everything that I do is in a dry lab, and we use computers to do all of our simulations. So classical molecular dynamics works by starting with, say, a molecule or a molecular system. Each atom is categorized based on the element and the other atoms it's bonded to. So in order to figure out how that molecule is going to move over time, we need to know the velocities of each atom. And this comes from integrating Newton's equations of motion. So velocity we can get from acceleration, acceleration from force, and force from the negative gradient of the potential. So you'll notice that the potential is negative in this equation, and that just means that all the atoms are going to move in such a way as to minimize their total energy, much like a ball rolling down a hill. So then to calculate this potential, we use a set of parameters, which we call force fields, that for each atom describes its bonded and non-bonded interactions with neighboring atoms. So then we apply these calculations to allow us to capture atomic level motions over time to produce a simulation. And basically we get out this really cool movie of all the different atom coordinates over each time step. So for part 1, we'll start with the air-sea interface. I mentioned earlier that CASE is really focused on getting the ocean conditions right in order to study sea spray in the lab. The ocean is in a constant state of phytoplankton bloom and death, and every sea critter is tangled up in this so-called biochemical engine. Organic material such as lipids and proteins and sugars accumulate at the ocean's surface and form what's called the sea surface microlayer. This layer forms because the organics are hydrophobic and separate out of the water the same way the oil and water separate. So when waves break, this microlayer is disrupted by bursting bubbles, causing organics and salt water to be ejected into the atmosphere as sea spray aerosols. So here I'm showing you a simulation of a fatty acid monolayer. Fatty acids are one of the most common organic surfactants found at the sea surface microlayer. And just as a refresher, a surfactant is anything that's surface active. Surfactants are amphiphilic, which means that they have a both polar and a nonpolar part. So when they go to the surface, they're going to orient themselves into what we're calling a monolayer, where the polar head group, shown in red here with the oxygens, faces the water, and the nonpolar tail faces away. And how these monolayers align actually impacts their climate relevant properties. So another kind of surfactant we find in sea spray is a phospholipid called DPPC. This one has two lipid tails, so it takes up a bit more space, has a bit of a larger fingerprint. And the head group is a Zwitter ion, meaning it has a positive and negative charge. So for this first phase of my research, I was really interested in understanding how adding complexity to these surfactant monolayers impacted their properties. So this green blob you see here is an enzyme that's common in sea spray called lipase. And it's actually another kind of surfactant, having both a nonpolar and a polar region. So one of the things we learned when we added lipase to the simulation is that it embeds into a surfactant monolayer and actually decreases the ordering of the surfactants around it. So you can see here that these DPPC molecules are curling up around the surface. And if you're really observant, you can also see that this disrupts the surface enough that it allows water molecules to permeate, which is entirely different from the tightly packed fatty acid monolayers, which are almost completely impermeable to water. So I've done quite a bit of work on interfaces with different levels of complexity over the course of my PhD. Here are just a couple of journal covers I illustrated. But for the sake of time, I want to move on to some of the more interesting stories in the next two parts. So from the first day that I joined Romi's lab, simulating an entire sea spray aerosol was kind of like my dream project. And in 2019, I applied for and received a generous allocation of time on the Blue Waters supercomputer, which actually enabled me to pursue this project. So just to put this project into context, in terms of experimental techniques, they're able to visualize aerosols and other sorts of particles down to about 30 nanometers or so, using different kinds of microscopy. But these methods are really limited in their resolution, and oftentimes just collecting the sample destroys its structure. And on the other hand, just as I've shown you, we've been able to use computational modeling to probe very small model systems, very simple systems. And these can contain, maybe up to 100,000 atoms or so, but typically not much bigger. And this is simply because our computers are just not powerful enough to perform all of those calculations. So when we got time on the Blue Waters and Frontera supercomputers, we were just really excited because these are some of the most powerful supercomputers in the world. And it's only because we have access to them that we were able to even think about simulating sea spray aerosols, because these have millions of atoms. And this has really enabled us to push all atom molecular dynamics into even larger timescales. So hi, this is me. And this is me bridging the two levels from simple systems to fuller complexity. So essentially, we just wanted to use molecular dynamics as a kind of computational microscope to study these systems that are too small to be seen with conventional experimental techniques. We were driven by questions as simple as, what do these aerosols even look like? What shape do they take? And how do the organics distribute throughout? And how does chemical complexity impact the overall dynamics? There are a couple of ideas about what these particles look like. The most dominant morphology is called the core shell morphology, in which the organics actually move to the surface, just like they do at the sea surface microlayer. Other models have been proposed, but essentially all of them involve complete phase separation between the organics and the water. But realistically, we don't actually know because we can't really see them. So this is a really data heavy slide, but I'm just going to walk through it with you. The first thing we needed to do for this project was build the particle and decide what kinds of molecules to put in it. So we combed through tons of experimental data and estimated that, submicron sea spray contains about 50% water by mass at 70% relative humidity, with the rest of the mass being composed of organics and a little bit of ions and such. So the experimental design, the game plan, was to ramp up the chemical complexity of the organic fraction in a series of steps. So I ended up building 3 different types of aerosols. We have a simple system, which I've labeled B, in kind of means basic or base system, where the organic fraction here is composed of only saturated fatty acids that we know are in highest abundance. And then the water just contains sodium chloride. Then, keeping the mass fraction of organics the same, I add lipopolysaccharides, or LPS, which are just a component of bacterial cell membranes and are also known to be found in sea spray. We also increased the cation complexity at this step to reflect the full cation complexity of seawater. So this means we just added calcium and magnesium and potassium. And then in the last and final step, we added glucose and a glucose oligosaccharide to the mix. So each of these aerosols contained about 3 million atoms, and they had a diameter of about 40 nanometers. I ran them in triplicate. Each of these systems were run in triplicate for 500 nanoseconds to a microsecond using the NAMD molecular dynamics engine with the CHARM 36 force field and TIP3P water model. So here's what happened. We initialized all of our simulations with the ingredients randomly distributed and then allowed them to self-assemble. To help you get your bearings here, these orange wormy guys are the fatty acids. The blue points are water, and those green blobs you see floating around, that's the same lipase we talked about earlier. On the left side here, I'm showing the first 50 nanoseconds of our simulation. What we notice almost immediately is those fatty acids aggregate super quickly. They start to form monolayers at the surface, or they go to the interior and form these oil droplets. After about a microsecond of simulation, we can see that the surface has become fully saturated with fatty acids. But one interesting thing to note is that this surface is not entirely smooth. It's tessellated almost like a soccer ball. These monolayers like to be planar, and they meet at these odd disordered angles. And then another thing you notice is that the lipase sits there at those intersections. And if you recall, lipase actually disrupts that lipid packing. So what it's doing here is it's probably helping to mitigate the stress on those monolayers caused by the high particle curvature. Then when we ramp up the complexity by adding LPS, which is this really long purple molecule, we see similar behavior compared to the simple system. You get fatty acids accumulating at both the surface and the interior, but now you can see the LPS stretches between the surface and the interior of the oil droplet. And you can also see the same sort of tessellating pattern happening with these monolayers. And then you see LPS and lipase both inserting at the edges. So we can clearly see how these aerosols are evolving over time, but I really wanted to figure out how to quantify these results. So I started with characterizing the evolution of the particle shape over time. We used 2 different parameters that essentially just describe the particle symmetry. I'm not going to go into the calculation details of that right now, but I wanted to point out in both of these plots, these blue lines really stand out. And each of these blue lines corresponds to the most basic system. So 2 out of 3 of the replicates evolved into a less spherical, more ellipsoidal shape. So I'm showing here one of the basic systems that adopted a large shape change, and another system, another base system that, remained relatively spherical. So you can see that these systems both visually align well with the data. So that's always encouraging. We also quantified the aggregation of lipids using a clustering algorithm. So up here on the upper left, I'm showing the fraction of lipids in the simulation that successfully aggregate into an ordered cluster. And we show that this aggregation actually happens quite rapidly. Everything seems to aggregate and stabilize within about 200 nanoseconds. So, and then on the right here, I've also plotted the number of clusters that form over time. So a lower cluster number just means that those clusters are merging. It starts out with a high number of clusters, and then they start to merge together as the lipids pack in a line. And then each individual cluster actually grows. So then investigating this lipid surface coverage a little bit further, you'll recall that earlier we noticed LPS and lipases inserting in between these lipid rafts. So we asked experimental collaborators if they could see anything similar in their experiments by adding lipase or LPS to a fatty acid monolayer. So the Gracian group was able to use BAM imaging, which is just a fancy kind of microscopy. And they were able to show that both LPS and lipase do in fact insert into fatty acid monolayers. But interestingly, they induce sort of these unique morphological changes. And this really requires a bit more investigation. We're not exactly sure what's causing these changes. But the fact that they do insert and disrupt the monolayer is in line with our observations from the simulations. So these results are encouraging as we move to start validating our simulations with experiments. And then finally, we wanted to address how the organic material distributes throughout the particle. So in order to do this, I subdivided each particle into concentric regions of equal volume, just like this diagram here. And then I extracted the atom coordinates in each section and calculated the mass fraction of the organic material within each volume. And I evaluated this for both the organic material, which is represented in this plot on the left, and then I separated out the lipases in particular, just for fun. The most important takeaway here from this plot is that you can see that organics go to the surface as expected, but they also largely hang out inside, which is pretty in line with what we were seeing in our simulations. So rather than fully phase separating, this happens in every single system, regardless of complexity. So this work really points to a new way of thinking about phase separation in sea spray. We believe that, rather than becoming fully phase separated, the surfactants and the high concentration of surfactants within the organic fraction leads to what I want to call a stabilized microemulsion within the particle. It can even leave parts of the surface exposed to the atmosphere, meaning that it's just water exposed. And this facilitates, water absorption or evaporation, as well as the transport of other atmospheric gases across the interface. So the bulk of my work has been focused on sea spray, but I've been involved in other studies. It included some other cover art here. I'm actually going to touch on this lipopolysaccharide paper in a bit. But yeah, this has been a really cool project to work on. moving on to part 3, I'm going to show you how we were able to apply the lessons we learned from simulating sea spray to tackle the coronavirus. So the Amaro group has been laying the foundations for large-scale virus modeling for years, largely focusing on influenza. So it was not really surprising when, last February, Romy came in and was like, hey, Abby, I think we should simulate the coronavirus. And, I was like, cool, that's awesome, let's do it. And I had started branding myself as sort of a large scale lipid modeling expert. So I had the opportunity to help out on a few of the influenza projects, just analyzing the lipid bilayers. And that really ended up preparing me for building these bilayers for the coronavirus. So I was actually in charge of building the coronavirus lipid envelope, but I, learned some lessons from the influenza projects. And it's actually really hard. It's actually really hard to build viral envelopes, in particular spherical bilayers that are stable. So some known issues with the influenza models are, in these models, the membrane consisted of only one kind of phospholipid, which I'm just going to call POP-C. And in reality, POP-C only comprises about 50 to 60% of the membrane. The second issue is that the membrane wasn't actually stable at all. It was so unstable that they had to actually restrain every 10th lipid during the simulation to prevent it from just completely breaking apart. So right off the bat, I wanted to build this new coronavirus model with the full range of lipid complexity, and also make sure that the lipid envelope was stable enough to run without constraints. So to determine what lipids to include, we needed to understand where the virus is formed within the host cell. The coronavirus actually buds off of the ERGIC, which is this region between the endoplasmic reticulum and the Golgi complex in our cells. This region has a specific and known lipid composition, which we used. Notice here that the POP-C makes up just under 50% of the total composition. You can also see that we've got about 15% cholesterol, and that cholesterol ends up being really important for replication. I'm not going to talk about that, but cholesterol is really, really important here. So once I was satisfied that these lipids represented a realistic coronavirus model, I was able to start building the envelope. So essentially, the Amaro lab had developed, back in 2014, this software called Lipid Wrapper. And this is what they initially used to build the influenza models. Well, it was built, it was coded so long ago that, the Python was way outdated, and I had to update it to Python 3. And then it wasn't accepting the more recent spherical mesh models that you built in Blender. And so I had to do a little bit of work on the software itself before I could actually apply this to our systems. But once I got that down, I was able to build the bilayer, and the process for this is actually you create an equilibrated lipid bilayer patch, and then the algorithm takes a kind of like a triangular cookie cutter shape out of this bilayer, and then it patches it onto this triangulated spherical mesh that you have to supply it. You can build it in Blender. And then it iteratively resolves lipid clashes all over the surface of the sphere. So we were able to end up with this nice, very cute spherical bilayer. So I was very excited about this. It was really, really big, had like 300 million atoms, which is 100 times bigger than the sea spray aerosol models that I was just working on. And so I built the thing and I pushed it up to the Frontera supercomputer to equilibrate it. And here's what happened. Here's the results. So you can probably tell that something's not quite right here. What you're seeing is the formation of holes. And this happens because those lipids like to be packed nice and snug. And lipid wrapper, during the process of building this sphere, ends up actually taking a lot of the lipid density away when it goes through to resolve the lipid clashes. So I needed to figure out how to solve this problem. So I developed this complicated workflow in which, and I'm just going to point you to these images here on the right, A, B, and C. I essentially just took our spherical bilayer and I allowed it to equilibrate, as in part B. So in the equilibration, it forms these holes. And so then I offloaded the bilayer and I patched it with new fresh lipids. And then I started the process all over again. I resolvated it and I pushed it back up to Frontera and allowed it to re-equilibrate, allowed it to form new holes, and then patched it again. So I do this over and over and over again until the system reaches an equilibrium lipid density. So you can see here that I ultimately put the system through 14 rounds of patching. And this took like 6 or 7 months, okay? This is a huge, huge system. And so the whole process was just so tedious and so slow. But ultimately, it was worth it because this work was part of a larger collaborative project that won last year's Gordon Bell Special Prize, which is a very prestigious award for setting new high watermarks in high performance computing. So with that momentum going, we couldn't just stop. So over the last year, I've been able to create an improved, highly stable lipid membrane that can run unconstrained, which was my original goal. And we've been able to fine tune it to reflect the Delta variant of the coronavirus by adding, Delta specific mutations to the spike protein. And then you'll also notice that we added these structural membrane proteins, which were absent from last year's model, mainly because we didn't actually know what those looked like. We didn't have the structures yet. So this is really sort of cutting edge research that we're working on right now. So just as a summary, the newest model that I've created is the model of the Delta variant, and it's largely improved over last year's coronavirus model. I was able to refine the workflow so that the full envelope stabilized in only 6 rounds of patching. We were also able to improve the accuracy of the whole model just by including the structural membrane proteins and including the mutations on the spike and so forth. We also, if you recall from last year's model, the membrane actually shrank because the lipids like to compress. So it ended up being a lot smaller than what was observed for the coronavirus particle. And so this year I was able to build this new envelope that was actually within observed diameters. So it's been really cool to work on this. But of course, the story isn't over. In May of this year, the CDC finally acknowledged that the coronavirus is an airborne threat, which brings me back to where I started, as promised, with aerosols. I had spent years developing methods for simulating aerosols as well as viruses. So naturally, I was very interested in quite literally combining my skill sets to simulate a respiratory aerosol. Because again, no one knows what these things look like. We can't image them very easily. So these particles, or just thinking about these particles, it left me wondering, how on earth does the virus remain viable when it's airborne? The atmosphere is actually incredibly destructive. If that virus diffuses to the surface, it's most likely going to get oxidized. It's going to denature. It's going to suffer from, solar irradiation or some other kind of gruesome death. So what's protecting it? To answer that question, we need to consider that these aerosols originate in our lungs. So how do our lung cells protect themselves from the atmosphere? The answer to that is pulmonary mucus, which is cute. Okay, so our science question really evolved into, what role does pulmonary mucus play in the airborne transmission of SARS-CoV-2? To start to address this question, we need to understand mucus a little better. Now, I know this is a very busy slide. I'm just gonna point out the specific parts of this that I want you to focus on. So over here, hopefully you can see my mouse. You can see that mucus is composed of these long strands of peptides, and these peptides are actually covered with sugar. These peptides are highly sensitive to the presence of cations. So calcium in particular causes this really pronounced change in mucus that makes it solidify, essentially. It makes it get all nice and compact and solid. It forms just this solid gel-like goo, okay? So this process is actually implicated in respiratory diseases like cystic fibrosis. In the presence of sodium, though, on the other hand, mucus transitions back into this fluid phase that can be secreted out by our cells into a protective layer. So, I saw this behavior, I'm reading about it, and I thought, wow, this reminds me of something I've seen before. If you recall, like I've looked at other molecules that are long and sugary. And so this reminded me of LPS. Lipopolysaccharides are also really long, sugary molecules, and they also exhibit unique phase behavior in the presence of calcium. So I didn't mention this paper. I didn't talk about this paper, but this is another project I worked on that was very, very cool. And I was able to show that calcium causes morphological changes in the LPS. And it changes the shape of aerosol particles that have LPS in them. So what happens here is that when you add LPS to the system, it causes these molecules to crunch up. And they actually kind of bind together, and this really prevents the diffusion of water, for example. And so calcium itself, like the hypothesis was that calcium is forming this divalent cation bridge between individual LPS molecules, and it just kind of makes them stick together. And this motif actually came up a lot in my research that I had already done. This is another project that I worked on in which calcium actually sort of acted as a glue. It glued this other sugary molecule called alginate to an Air-C interface via these cation ion bridges. And this calcium ion bridge, this motif, is really, really strong. It's a very favorable interaction. So these things really, they stick together. Calcium is like this great aggregator of sugars. So I'm sitting here thinking, okay, like maybe calcium is important. And so these hypotheses really drove the development of the respiratory aerosol project. I predicted that mucins, these sugary molecules, form a protective cage around the virus. And they do so using these calcium bridges in which they, which can connect the mucins together, cause them to aggregate to each other, as well as connecting them to the spike protein at the surface of the coronavirus. And the spike protein also has these sugars attached to it. So the mucins can interact with those sugars as well. Mucins are also going to increase the particle viscosity, which is going to impair water loss and prevent the reactive uptake of atmospheric acidic gases, for example. So all of this is serving to keep that virus alive long enough to infect the person next to you. So I present to you the first glimpse of a billion atom submicron virus-laden respiratory aerosol. This work was a finalist for this year's Gordon Bell Prize and represents another paradigm shift for computational virology just by trying to understand the virus from an airborne perspective. I can't tell you how much work went into building this, but it was a lot. It was a lot of work. And I was so relieved when we loaded it onto Summit, which is the second most powerful supercomputer in the world. And it ran just on the first go, basically. And that was just really validating because I'd spent all of this time. We had like one shot to load this structure onto Summit, and it was successful. And I just spent all of this time making sure that, you have to make sure that the atoms aren't overlapping, to make sure that there aren't any clashes, that they're not too close together. Otherwise, the energy just destabilizes and the whole thing will break apart. So this is really a significant accomplishment in my opinion. A billion atoms, that's just unheard of. So while the COVID-19 pandemic has been just enormously devastating on all fronts, it has really brought the scientific community together in so many beautiful ways. And I've had the opportunity to work with amazing scientists all over the world to address the most pressing scientific challenges. Our work has been featured in the New York Times twice now, which is just a huge honor. And it's really cool to see computational chemistry getting attention that it deserves. Actually, as you've heard before, as Romy told you, the most recent New York Times article is out in print today, which is really brilliant timing. And I love it. So in summary, throughout my PhD, I've shown that increasing the chemical complexity at interfaces impacts climate relevant properties on the molecular level. I've contributed a paradigm shifting perspective on the morphology of sea spray aerosols through large scale molecular simulations. And I've improved simulation methods for whole viruses in atomic level detail, and have successfully created a fully stable viral envelope model that can be fine-tuned to reflect new coronavirus variants as they emerge. And then I've also provided the world's first glimpse into a virus-laden billion atom respiratory aerosol. So just to show you, this is just for fun, just to show you how, we've really been able to ramp up the complexity and push molecular dynamics to just new levels. And so I'm showing you all the different scales that I've worked on. Remember our lipase that we started with at the very beginning of this talk? Now we have like 8 of them in here in our sea spray model. And then we scaled up to 100 times that size by looking at the coronavirus. And then, as if that wasn't enough, we scaled it up to 1 billion atoms, which is currently it's one of the biggest systems that have ever been simulated with all atom molecular dynamics. And so more powerful supercomputers are incoming. And so, who knows what's going to happen next? I'm sticking around as a postdoc just for a little bit. So, maybe we'll find out. Okay. And with that, there are just way too many people to acknowledge. All of my projects have been highly collaborative, and I couldn't have done any of this without all of these humans that I've listed on this slide, and all of you here who've come to this talk. My PhD experience has just been better than anything I could have ever dreamed up for myself. And I've just worked with so many amazing people. And I have to point out, I have to thank Kim Prather, because I would not be here without her. Like, she, right from the very beginning, from the first day of grad school, she gave like a 20-minute faculty research talk. And from then on, I was just like, all right, I study sea spray now. That's just the thing that I do. And Romy Amaro, she's my advisor, and she's just built the most amazing and talented group of humans I've ever worked with. And she's led us all into these crazy projects with so much love and ease. And I also I want to thank everyone on my committee and everyone watching. I appreciate you all so much, and thank you for being here. And I need to thank the NSF. Of course, NSF has provided the majority of my funding, and I'm eternally grateful for that. Okay, and I will take questions if you have them.
That was great, Abby. Thank you so much for a whirlwind tour of all that you've worked on.
Buckle up.
It was fantastic. You did. So I don't know if folks, I know there's some, there are some questions in the chat, which I can read out, or people can unmute themselves and ask, or you can raise your hand and I'll call on you. I see Fiona has her hand raised. Fiona, do you want to go ahead and ask a question?
Of course. Beautiful talk, Abby. Just phenomenal, show-stopping, but you already knew that. I was going to say that. My question is, because I know that this is, just a kind of an overview, but was there any story in this, in your time in graduate school, like from a research perspective, that you had to leave out, but you wish you could tell? Any other little bits, maybe with the LPS or anything like that? Just any extra story.
Thank you for that question. I was actually, I was pretty proud of the LPS story. Now that you mentioned it, I feel like that project really, it was one of my first projects where I felt like I had a little bit of ownership over it. And I feel like I learned a lot. And so I feel like, the story is interesting, but from like an emotional perspective, I felt like that's where I like became a scientist, and started figuring out, like what analyses I could use to solve these problems and really understand what was going on with the experimental observations we saw. So.
Thank you.
Great.
Loved it every second.
Sophia. Sophia, yes, there she goes.
Abby,
Hello.
Congratulations. This is amazing work. Can you hear me?
I can hear you.
Hey,
Thank you
can
for
you guys
coming.
hear me okay?
Yeah.
Hi, greetings from Greece. I'm in Rhodes right now. Yes, of course.
Oh no, she's breaking
I'm
up.
losing her.
I just want to congratulate you first of
Congratulating me for
all.
something.
You really pushed the boundary of
We might have
Sophie, we're losing
Sophie,
you.
you're breaking up.
Super,
You know what I
super
can
proud of you. I talk about you guys
So
and your work
congratulate me.
all the time here in Greece. COVID. Can you hear me all right?
No.
Yes.
It's pretty,
Okay,
it's
I have a
breaking up.
distance. I'll write my question in the chat.
I see your question about the boundary conditions. I can read that one. I'll go ahead and read that one. So she, as you heard, she says, super amazing. She's so proud of you. Question, what are the boundary conditions you're using in your
I
simulations?
put my question in the chat a little bit.
And then how do you determine them? Sorry, I'm a computer.
So this is a great question. It's actually it's one we're still trying to feel out. I'm assuming you're asking about the boundary conditions for the aerosols. Yeah, it's pretty difficult because if the boundary box is too small, then the aerosol will sort of evolve and start to interact with itself. And so you really need to make those boxes like extra big. And the consequence of that is, how if you're familiar with molecular dynamics, you'll know that we typically talk about performance in terms of system size, like number of atoms. But when we're looking at the aerosol simulations, it would appear that the performance is a lot lower. But actually, the way that the code works is that it scales with volume. So when you even if you keep the number of molecules the same, scaling up the box size just really drags down performance. And so it's one of the things that we had to figure out how to manage when we were doing this billion atom system, because now it's behaving as if it's 2 billion atoms, because even though it was 1 billion atoms, we had to expand the box size to just make sure that the particle is not going to interact with itself and change shape and so forth. So yeah, we're still really feeling out like exactly how much bigger the box needs to be. But yeah, that's a great question.
Thanks for the answer and congratulations again.
Yeah, Abby, fantastic job. It's just so proud to read all your articles come out on Twitter and the
Hi,
Gordon
Greta.
Brett Prize. Yeah, it's amazing that I work in the same office space as people like you. But yeah, I had kind of a follow-up question to what Sophia was asking, that how do you even analyze such massive simulations? Do the traditional tools that people like me use, like Chimera, VMD, do they still work for such massive systems, or do you have to make your own stuff from scratch? And how do you ensure that your system is equilibrated, because there's so many atoms to look at?
Yeah, okay. So I'm going to take this into two chunks. One is the software chunk, like the logistical side of it, and the other is is it equilibrated question. So building the system with a billion atoms, I actually broke the code a couple of times, and we needed to work with the VMD and NAMD developers. Like I have them on Slack, like basically direct messaging them, like, I broke this thing, can you help me fix it? Because no one had ever broken it in this particular way. And so then building the system, I actually had to build it in chunks of like, millions of atoms, which can be visualized and handled in VMD. And then I had to use this thing called the software called PSFGen, which is NAMD software, and it is actually able to, and I had to get like a special version of it for our systems, but it's actually able to patch all of these small bits together into one structure. And, you know, even now, like I need a special GPU just to load this this giant structure into VMD. So it's actually a lot, it's a lot more effective to break the structure into bits and analyze them separately. And yeah, that's basically what I had to do with the aerosol, with the sea spray aerosol simulations as well. And then your question about is the system equilibrated? You know, this is like one of the things that I was very concerned with in the very beginning, just because you know, we could only run this for 500 nanoseconds to a microsecond, which is it's not really long enough. It's not long enough for what I would want. But then one of the things that I was thinking about, and one of the stories that's kind of emerging right now, in terms of are these aerosols actually even equilibrated in ambient conditions? And we're starting to think like probably not. Like they're being ejected from the ocean. They're being processed by enzymes, for example, that we have found to retain activity even when the particle is airborne. You know, it's constantly reacting with sunlight and atmospheric gases and so forth. So these particles are very dynamic, and it makes me think, you know, maybe we'll never reaching equilibrium is not really like the goal. It's just we just really want to understand, we just really want to gain some insights into how the particle is evolving.
Okay, I don't see this question in the chat, so I'm gonna just ask here. So you said that you're gonna stay on as a postdoc for some time. What's next? What's the next headline for New York Times from you?
That's a good question.
Don't spoil it. Don't spoil it.
I'm not gonna spoil it, but okay. I will tell you that the billion atom particle, we got it to run, okay? Like we got it to run unconstrained. Guess how many nanoseconds we got? 4, okay? 4. We got 4 whole nanoseconds. Like which is amazing. Like it's amazing that we got 4 nanoseconds with this system size. But like yeah, we obviously like we didn't see any of the lipid aggregation that we saw with the sea spray, and this is what I'm really interested in seeing. I want to see how those lipids aggregate and what happens with the mucins, and I want to like find out if my hypotheses were correct. And so that's really, I hope that's the next phase. That's the Abby postdoc phase, is hopefully going to be getting these supercomputers to agree to let us continue running these giant systems, and maybe even you know, actually like digging into the calcium hypothesis, for example, or digging into like, you know, is the Delta variant stickier to mucins than other variants? So there's lots of different avenues that we can take, but yeah, I'm hoping that's the next step.
All the best. Thanks so much.
Thank you.
How about Gil? I see Gil McHugh. Thank you, Kartik. Great questions. Gil?
Hi, yeah, thanks. Abby, I just wanted to add my congratulations. That was just beautiful. And as you can see in the chat from Kim later on, I think that every case presenter wants a copy of your slides. They were.
Oh yeah, absolutely.
That would be
I have
great.
them all.
It really shows the power of images, particularly when narrated with such clarity and insight. So it was really great. I wanted to ask you a particular question about the lipid, the lipase and the LPS come to the surface of the aerosol particle in sort of the gaps between the tectonics. In Mon and Kyle's studies, they were done on flat surfaces.
Yes.
So how do you, but you pointed out, you thought that they went to the intersections between the plates to fill in, so maybe to reduce stress on the curved surface. Did you also see them come in within the I'm sorry, the fatty acid blankets on top, or did they only come to the intersections between them?
That's that's awesome. That's a great question. I love that question because I was looking at this in detail. Lipase tends to not embed into those monolayers directly. Lipase tends to more insert at the intersections, and I think that's just because of the way that the structure of lipase is. But LPS, on the other hand, it has that really long sugary head group, but then it has these like 6 palmitic acid like lipid tails. And you can actually, I wonder if I can, I'll show you, because it's like one of my favorite things. I'll scroll back. It actually embeds like really nicely into these, into the monolayers. Where's that image? I have to scroll back so much. Da, 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 da. It's one of the last ones. Here. Do you see the purple here?
Yeah, I see. Oh, oh, that's interesting.
It inserts right. It goes both to the intersection here, but it inserts almost seamlessly. Like you almost don't notice it, but it's like seamless how these order up with the fatty acids around it. And I mean, I just assume it's because they're palmitic acid like. They're saturated hydrocarbon tails with the same length, the same tail length. So they just kind of incorporate.
Is the density within those regions, is that at the sort of the equilibrium surface coverage density that you might get from a pure fatty acid? So it really is well packed in those. So if you made this argument, a beautiful argument at the end about going up in length scales up micron. Do you think that the bridges that you see in the aerosol particle, is there a scaling argument that tells you where you will stop seeing bridges, like at what diameter of the particle it will phase separate into the conventional picture? I think this microemulsion picture is just gorgeous, but do you think it will survive to larger scales?
Hold on one second. I've like lost my screen. Here we go. Okay. So your question is about the phase separation and whether it scales up to larger particle diameters. This is really one of the questions that I've been very interested in. We've submitted a couple of proposals to build, larger sea spray aerosol models. But I can tell you that we've actually started to see evidence in recent literature that like organic inclusions can be found in some of these particles. And also, I don't know if you saw this. This was way back when Joe Patterson was working with CASE, and he did that cryo electron microscopy study where we found the whole bacteria and the whole viruses that actually get incorporated. In that same study, I recall I was like sitting there and looking through his cryo-EM images, and you can actually see little vesicles, little lipid vesicles that are dispersed throughout the particle. And I feel like that didn't really get a lot of attention in that paper, but you see it in those in the large scale. So, and it makes sense, right? Because, surfactants form bilayers.
But that will be an equilibrium question, right? That's going to really test. I mean, the aging of the microemulsion. Yeah, this will be absolutely fascinating. Yeah, this is fantastic. Thank you. This is
Can I chime in real quick and remind you of something, Abby?
Yes.
Also, we did see it in the reactivity study. We saw evidence that the reaction was slowed down and that the morphology of the particle actually changed significantly. We could see it in the shape, which was shocking to me, that the actual size of the particle changed because of the weird shape that formed when it probably changed phase, which is pretty, and that's a big particle. Those are micron sized particles, right? That's what I mean, it's a stretch to say it, but boy, we saw the shift. So you've seen it, I think, at the larger scale.
Thank you, Kim.
Great. And then another Kimberly.
Sorry. Hello. Okay.
Hi, Kim.
Fantastic presentation. I super enjoyed it. And also I really enjoyed the Musin story. That was super interesting. But my question is actually more related to the microemulsions again. Sort of thanks, Gil, for leading that. But I was curious, I don't know if you have like enough, or if you've like looked into this at this point, but I was curious if the composition within the actual little microemulsions changes as a function of like the size or the diameter of those like pockets.
Yeah, that's a good question, and I'm not sure. Just because, you know, I even kind of was talking to Romy about this. I even hesitate to call that a microemulsion because the particles are so small. I'm not exactly sure what to call it. We would need to look at bigger sizes. And another thing that I learned just in like reading the literature is that emulsions don't just have to be like oil and water. They can contain even just like varying levels of solubility. And so you can have like intermediate solubility like bits within an emulsion. And so the microemulsion is actually just what it just means is that it's like thermodynamically stable. And so it's just not gonna separate out ever, like even if you equilibrate it out long enough. Yeah, I don't know. Future
That's
work.
super cool. Thank you.
And then Lane, this is so
Lane?
great. We have so many questions.
So many questions.
Yeah, just as everyone said, thank you for a fantastic defense presentation. It's very impressive. And one of the most impressive things about it, and I think everyone agrees, and I've always felt this way about your work, is your ability to make excellent graphics, like very nice pictures of molecules and plots. And so this is something I'm certainly hoping to get better at. And I know that there's lots of other people who would be too. So what advice do you have for us who want to make nicer graphics?
Yeah, so pick Lorenzo's brain a whole lot. So advice number one, because he knows the inner workings of VMD and making VMD make cool images. So that's the first thing. But the second thing is I actually I'm an artist by training. I went to WashU originally to be a fine arts major. And so like I've grown up around like, you know, taking art classes. And so I have a good feel for like composition and balance and color theory and all of these things. So, you know, reading up on, you know, even just like learning about photography and how you set up a composition of a graphic to make it balanced is something that I found really useful. Like
Wow.
I can look at a you know, an image and just be like, okay, it doesn't feel right. And here's what you can do to make it like look more dynamic and balanced.
So maybe take some art classes.
Yeah.
And you use VMD, you don't use Blender for your graphics, or do you use Blender for some of them?
I have not used Blender. Nick really likes
Just
Blender.
VMD. Okay. That's
Really,
good.
it's a big learning curve, and I just haven't had the energy to look at it yet, but it can make really beautiful things too.
Yeah. And for your plots, do you mainly use Matplotlib?
Yep.
Okay, so you just get really good at that, like just really learn everything you can about that, or maybe just sort of, I guess, be willing to put in the extra time and effort to make really nice plots, make them perfect.
Yeah, I mean, yeah, Matplotlib is my best friend. Yeah, and then there's a little bit of like, I build them all with PowerPoint. It's very sophisticated. So
Well, that's good. It doesn't sound, well, I guess I should say it sounds accessible. to
Yeah. Oh, and then the other thing is with Matplotlib, you have to set the font size to like a like comically large.
really? Okay.
Make it as big as possible, and then set your like graph, your figure size to like comically small, so that you get like a nice like text to figure ratio, so it's like
Wow.
readable.
That sounds good. I appreciate that tip. Pro tip.
Yeah, no problem.
Great job, Abby.
Thank you for the questions.
Yeah, those are great questions. And I just want to add, if I can just say too, especially with the coronavirus images, there's been, Abby's work has obviously been super central to that. But as she mentions too, that's been really an enormous amount of teamwork by, a lot of other folks in the group too, including the use of Blender. Actually, Nick did some beautiful visualizations that sort of mixed Blender with VMD as far as I understand. So really neat. Anyway, Chris Lee. Hi, Chris. Another phenomenal graduate.
No, I think we've been clearly left behind by all the amazing work that Abby and others have been doing in the lab. Really astounding work and really nice presentation about all of it. I thought that your point about the non-equilibrium states of these aerosols was very interesting. And I suddenly realized that I have no idea how to even define the temperature of an aerosol system. And so presumably it's like body temperature when it's spat out, but then it's such a small droplet, it probably cools very rapidly. And so I'm wondering, how did you decide what temperature to simulate the system at? And does the temperature change, or how do you wrap your mind around some of these questions when simulating these ultra complex systems?
Yeah, that's a great question. It's actually like something I've been really uncomfortable with since joining, because you learn that the temperature of these particles drops just so much as it moves higher up in the atmosphere. But the thing is, like, the force fields just aren't optimized to handle the temperature change. So all of our simulations are done at 298K, just sort of ambient temperature. And that's just because our force fields are optimized for that. And I think it's still valuable. We can still get insights out of it, and there are other groups that are working on looking at like ice templating and ice nucleation, and they're also developing like special force fields to get those parameters right. So yeah, it's not the best answer to that question. It's really just the limitations of the force fields right now. Otherwise we would go colder, I think. But then on the other hand, having the system heated up makes it go faster. So makes it equilibrate faster. So there's like some benefits to doing it that way.
That was my other question, is that, oh, go ahead, Romy.
Well, I was just going to say, and I think, Abby, the day that that evening when you were getting it loaded onto the launching pad at Summit, remember, I think we had like a moment where we thought, well, if we only get one shot to run this, what should that temperature be? And I called Kim because I was like, Kim, if we only get one shot, what should it be? And we just went with, sort of like the ambient, like you said, but it was a bit nerve-wracking. Anyway, sorry, Chris.
Yeah, because we had it at 310, which is what we had simulated the coronavirus model. Yeah,
and we had to drop it.
Yeah.
Yeah, that's awesome. It's interesting to see that there are these different limitations from the force field and other areas that kind of hinder the model development. I'm also curious, maybe the approximation isn't so bad because we don't care about aerosol particles that go up into the upper atmosphere. We're caring more about it in kind of maybe crowded spaces, and which are also a bit warmer because they're packed with lots of people, like around holidays or something.
Yeah, especially in terms of the respiratory aerosol. Yeah.
Very cool work. Thanks very much.
Thank you.
What a lovely set of questions. We really, she's already had about 30 minutes of interrogation by the group. This is great. Are there any other questions before we break into the private session with the committee?
Griff?
Yeah, yeah.
Oh yeah, sorry.
Hello, can you hear me?
Put his hand up.
Yes.
I can hear you.
Yeah. So it seems to me, forgive me if this has already been answered. I had to step out for about 20 minutes a while ago, but there's like there's a confluence of kind of three skill sets that happened. I'm wondering if you saw this in advance, where it's like, okay, you're an artist, you're interested in aerosols, and then you're also this computational chemist, where it's like, wait a minute, I all of a sudden have this unique skill set where I can bring a model of this to be seen and shown and represented in a never before accomplished way. Was that kind of quote unquote by accident, or did you have a moment where I'm like, I'm going to go for this? Or did you have this eureka moment where it's like, wow, I think I'm put on earth to do this?
That's such a nice question. I think it's funny. Actually the reason I switched from fine arts to chemistry in college was because I came in and I was like, all right, I'm going to be a biomedical illustrator. This is just the thing that I'm going to do. And I had to take biology, or I wanted to take biology. I wanted to get a biology minor. And I called up the school and I was like, I want a bio minor. What classes do I need to take this fall? And I'm like an incoming freshman. And they were like, oh, well, you can't take biology until you take chemistry. And I was like, I thought I would never have to take chemistry again. Thank you. But but okay, I'll take chemistry. And the first week of that Gen Chem class, they had us actually plotting the atomic orbitals in polar coordinates using the solutions to the Schrodinger equation. And I was just fascinated by like the way that you can visualize like mathematical equations. And it was very beautiful to me. And so I feel like my art brain was just like, oh, you have to pursue chemistry now because it's so beautiful. And it just makes sense. And, so I just kind of fell in love with it that way. And then I discovered computational chemistry like way later on. And it just was like this beautiful, like everything is so visual and you have all these opportunities to make these like beautiful plots. And so I feel like things are they just kind of fit together, and I've like found a nice groove. It's very fulfilling.
I think it allows your work, which can be seen on one level as being very lofty and for uber experts and everything, but it allows that work to also cross over to inspire and influence people. Like, say, my 10-year-old son Mason, I showed him your computer models and it just completely blew his mind. He was at the time doing a science report for 5th grade about Jonas Salk and the stuff that he's done in his work. And then I showed him your work and his eyes popped out and he was like, that is so much more awesome than any of the drawings, any of the diagrams, any of the things that he's seen either in the books or on the web or whatever. I'm like, this is what Abby is doing now. This is her cutting edge work. And it just blew his mind. So good on you for the excellent work with the graphics and getting that to a wider audience where people can see, and it makes it real and dare I say beautiful for them. It's an artwork that you're doing with this science. And so thumbs up.
Thank you. Yeah, it's a very important part of science communication. Definitely.
Yeah, absolutely.
Thank you for the
Just
question.
untouched.
Thank you for coming, Griff. It's good to see you. Griff is my tide pool friend.
Yeah.
That's so cool.
Yeah.
Well, great. Well, are there any more questions from the general audience? Okay, if not, I'm going to try to make a breakout room. Let's see how this goes.